Okay, so a couple of things. The library assignment or the information literacy assignment. I have a long list that I just got from the librarian of those of you who've actually attended one of the information sessions. She sent me an email that had a specific address, a specific fix for concerns and problems some of you had when you were trying to use Outlook and actually register for the webinar. So I copied her whole email and posted it in the announcement last night. So if you see that, there's a little tip in there if you're having trouble registering through Outlook with your Camden County email. So try that first before you just email her directly. The second thing is she does send me a list every week of the students who've attended. And because some of the other BIO1 instructors have asked for more sessions, she did put some more sessions on. So if you haven't attended and you still want to attend so that you know how to use Science Direct for the information literacy assignment, then you just need to sign up for one of those sessions. Okay, so I think she put 4 more on. I didn't look at it too carefully. I saw the email last night and I was like, oh, I'm going to put that up so I can tell them that tomorrow morning, first thing. I am keeping a list.
Your audio cut out.
You did.
Oh, never mind.
I saw it. I was trying to mute somebody else. Sorry. I'm keeping track, not because I'm giving you points for attending the webinar, but when you actually turn in the assignment, if some of you are completely way off the mark, then I am going to look and see if you attended and you at least kind of know what's going on. If you don't attend any of them, the point is not for me to go over the whole, I'm not a librarian, right? And I know how to use Science Direct, but I'm not excellent at teaching how to use Science Direct and teaching the whole information literacy assignment. So that is part of their job. And so as to not interfere with that, I'm not going to repeat the whole thing for you individually, one-on-one. So I don't have time. And we set up this webinar for you to go to. So please, you do have to register, I believe, 24 hours in advance. And I do see, she also keeps track of who registers and then doesn't show up. So I can see that some of you have made an attempt to try to go and then you forgot. Just sign up for another one. There's no consequence. You can sign up for, I don't want to say as many as you want, but you can sign up for more than one, but just try to get to one. It's really important, again, because I'm not going to go through the whole assignment again and how to use Science Direct and all of the resources of the library. So I do have a short video just going through using Science Direct for searching, but it doesn't include all of the other things the library has to offer and the librarians can help you with. So you need to attend the information literacy webinar as soon as you can. So that's one thing. Second thing about the elephant toothpaste labs, I haven't even looked at them yet. This is not an auto-graded lab, so I have to actually go in, download your photos to my computer, make sure that I don't have any viruses that you're bringing to me, so that you're going to crash my computer, and then upload them back and look at them. So please, please, please try a still photograph would be better than a video because I don't have the capacity to download all your long videos. Still photograph would be better, and please try to upload it in Canvas because then I don't have to download it all to my computer. If you send it to me as an attachment to an email, it's going to be graded last because I'm going to just make a list of those and put them off until I have time to actually sit down and go through them. If they get loaded into Canvas, it's much easier for me to grade. You will not see any score until after I've actually graded it. So I have had a few people send me emails over the weekend. I did the lab and I don't have any credit for it. You don't have any credit for it until I actually grade it. And then when I grade it, I will give you credit for it. And I have to manually grade it and then put that score into Canvas. That won't be happening overnight. As I mentioned, there's 85 of you. And so I would like to grade them pretty much all at once. They were due Friday. I had about 6 people tell me they couldn't get them done because they didn't have their lab kit or whatever. So I probably will start grading them today, start looking at them today. I won't be able to sit and look at all 88 of them, at one time. It's no different than if I was grading a stack of papers and there were 88 of them. I wouldn't sit down for 4 hours straight and just grade all of them. So those grades will show up as I complete them. So you just have to know that. Labs are going to be due every Friday. Remember, we talked about this every Friday for the next 7 weeks. So we're finished the labs, except with the exception of the Friday after Thanksgiving, because we'll be with family or doing whatever we do on that special Friday after Thanksgiving. So the next lab that will be put up, Professor Ramirez Wright did do a couple more videos. So I think the kidney dialysis lab is in there right now, but I might replace it. I don't think I published it yet. So I'll put something up today for Friday. Those are, please try to get them done by Friday, but if you can't get them done, I would say Friday's moderately hard due date. Monday is the firm due date, because I'll start looking at them the following Monday and I'll grade through the week. And if you haven't turned it in, then I probably, by the end of the week, I probably won't grade it until the end of the semester. So you're not going to get any deductions for late. I think so. I haven't looked, Leah. So that's what I just said. I haven't looked at any of them. If there's a problem with any of them, then I'll just send you an email and say, can you do it this way or that way? If that isn't, I think I left it open for as many different formats as possible for you to upload them. So I'm sure it's not, I don't know, I can't promise it isn't a problem, but it shouldn't be a problem, no. But I won't know until I actually look.
Can I ask a quick question?
Sure.
So with the lab, I put this as a comment in Canvas, but the instruction manual for the lab, it was obviously all in metric, but it didn't supply any measuring tools for the metric system. So I had to like
there was supposed to
convert
be
them all to household.
There's supposed to be a small graduated cylinder, 10 mil graduated cylinder in your box.
I didn't open the other lab kit boxes.
I would open the other boxes and look for one. I'm pretty sure there's a 10 mil graduated cylinder somewhere.
Yeah, because I just converted everything to tablespoons and teaspoons because I was like, okay, they don't have a graduated cylinder in here.
All right, I will make a note of that and try to ferret that out. I can't promise you that I'll have an answer. I was under the understanding for that particular lab, I don't think it really matters. Like it
wasn't like a super accurate lab, which is why
it's just approximate. Yeah, so I don't think it really mattered. Just mix a little of this, little of that, and watch it foam, right?
Yeah, but I was just like, oh, I hope this isn't a problem for
no.
It's more complicated.
No. These were all, I believe, I didn't choose them. The labs are always going to be due on Friday, Brianna. I didn't choose them. I wasn't part of that team of person who picked them, and that person doesn't teach BIO1, but anyway, that's another thing that I'm still angry about. And so my understanding is, and remember, I don't have one, like I didn't get a kit, so I don't know what's really in your boxes. My understanding was that everything you needed was in each box. I do kind of remember now, someone said the elephant toothpaste did not have all of the measuring devices. I do kind of remember that. These were converted, basically, we purchased them, and they normally are sent to a classroom in a big box with enough for 24 students, and they repackaged and repurposed them in the speedy manner as fast as they could during like July and August. So it's not perfect, right? And that's why I'm not really giving you, you're getting completion points for the labs, and I'm going to give you a lab quiz at the end, right? Because I can't really hold you to do these labs because I can't actually help you do them, right? So just do it the best you can. Just, yeah, you can convert it to teaspoons. 1 milliliter is about a teaspoon, 20 drops of water, 20 to 25 drops of water is about a mil, if that helps. Sort of rough measuring. But yeah, just give it a try and just photograph and answer the questions. It's more important that you just go through the motions of finding a place to set it up and doing it than the actual results for this particular one. The next lab, which I think we'll do kidney and blood filtration as our second one. It has a little bit to do with osmosis. And then we'll do acids and bases for the 30th. I'll just put the instructions, just like before, I'll just put the instructions up separately and then an assignment that you can upload a photo and answer the questions. As I mentioned, it's a lot easier for me if you send them in Canvas, if you upload them in Canvas. If you send them attached to an email, that's kind of a last ditch effort if you do that. And like I said, I won't be grading those first. I'll be searching for those later and you'll be like, I sent you an email, and I'll be like, okay, what day was it? Because I have 60 in my inbox today and I can't move them from my college email into Canvas. So I just have to grade it as an email and then delete it. So that's a little bit more problematic. If it's in Canvas, it gets kept. So that's that. That's all I know for lab, I think. Okay, you're having a quiz. Go ahead. You're having a quiz tomorrow, yeah?
Yeah, I have a question about the Pearson reading.
Yes.
Because right now it just says to read a bunch of chapters. There's no homework assigned.
For chapters 11 and 12, or sorry, 12 and 13, right? So those haven't had, those don't have the dates changed yet because they're specific to when we get there. And right, there's never any points associated with the reading.
Yeah, I know. So should we not worry about the homeworks for these yet?
For 12 and 13, no, I haven't built them out yet. So I was going to put due dates for the reading for 12 and 13 for tomorrow. But again, it's just, basic due date. And then the homeworks for 12 and 13 are not as extensive. It's looking at cells and looking at the stages of mitosis and meiosis. So I'll probably be building those out today.
Okay. And also it has reading chapter 13 as first and then chapter 12. Should we do that or do it 12 and then 13?
I do 12 and then 13.
Okay.
I have a question.
Yes.
For the skills and scatter plot things we have in Canvas, I noticed that the dates are still in June.
Yes, because we started with labs. So in the summer we didn't have any wet labs. We didn't have any kits. So those are exercises from summer, and I've been adding them in or not adding them in based on whether or not people had their kits and whether we had the supplies. So I had them built in there, and then I didn't assign new dates to them on purpose because I was waiting to see if most people got their kits and most people got their lab done.
Okay.
They're pretty quick, Leah. I think they're 15 points. So, and there's two of them right now, I think, hanging out there that have June due dates on them. So I will probably assign dates to them and slip them into the next module. They're they're free floating. I just use them to fill in when we don't actually have a lab, but it's a practice, like a math practice and a graphing practice. And I feel like it's valuable since we're not collecting data and making graphs in lab, which we normally would be doing.
Yeah, I noticed I did click on the assignments though, and nothing came up for me to complete it. So is that going to change once you change the dates?
Yes. Okay. So that's kind of how it always is in, give me a second here, I'm thinking. In mastering, if it has an old due date on it and you click on it, it won't take you to the assignment, right? You won't be able to complete it. Not until I put a new due date on it and sort of a start date and an end date and put it on the calendar. Then when you click it, will it take you there? So those June dates, I realize it's frustrating. I know it's frustrating for me too, guys. I'm trying to take I had a 16-week course that I had to teach 8 weeks of in the spring. Then I had to make it into an 8-week course for the summer session. And now I'm trying to stretch it back out to the full 16 weeks. And what I had built as module to module isn't always appropriate now for a longer semester, right? It's way too much to do like in a week. But if we were in summer, we'd be doing all this in 4 days because, or technically in a week because we only have 8 weeks. So that's why those dates are on there, right? Because it's left over from summer. They're all assignments. What happens in Canvas is they copy your whole course over, and then everything's just sitting there, and then you have to kind of sort through. So I'm kind of building it out into a 16-week course as we go along, and it's the best I can do. So, just try to be patient. If something, you click something and doesn't go anywhere, then it's not something that's due.
Okay, thank you.
Skip 10.4, but we had to read it on the assignment that said to read chapter 10. Let me see, I think it's a skip. I think the reading assignments in mastering don't transfer over as I had them. So 10.4 is a skip, yes. It's about C3 and C4 plants. And someone had asked that last week, I think, as well. And so, yes, 10.4 is a skip. I like that last picture at the end, though, that kind of shows the whole inner working. 10.5 is kind of nice summary to integrate photosynthesis and the last quiz. I need a little more vocabulary than that. So the mastering quiz or the last lecture quiz? Because the lecture quizzes are graded automatically. And the mastering quiz was graded automatically as well. Does mastering prevent you if it's past the due date to go in? I may have to give you an extension. So if it's the mastering quiz, send me an email in Canvas and I can give you an extension on that chapter 9 quiz. If it's the lecture quiz, then we have something else to talk about. And where was the other chat thing? Oh, and for InfoLit, any random biology topic that you're mildly interested in, I would try to pick something that is not, I would try to pick something that's not mental health related, because a lot of those, unless you find an article that is less psychology and more biology, I would like you to sort of stay away from articles where the data is all self-reported, meaning reported a person's like pain index or their feelings that day, or if they're feeling depressed or if they're feeling happy. So I would try to avoid those, mostly because the point of this article is to look at a peer-reviewed experimental, not observational science articles. So I would avoid those topics. And as we mentioned before, I try to avoid things that have a lot of computer modeling in them, unless you can explain the algorithm of the model. You are required in information literacy assignment that I give. If you look at the rubric, it asks you to describe one of the experiments in the research section of the paper, of the research paper. So the simpler the paper is, the easier that will be to do, because I would expect you to write it in your own words as best you can. If you have difficulties, if you run into trouble, just let me know. Try to reach out. You can send me your articles if you'd like. I can take a look at them. Just real briefly, even just the abstract, it helps. I can just say, yes, that's the right kind of article. That's a great paper. Whatever. That's pretty common. People ask for help. So I would pick something simple if it was me. I would pick something that I'm interested in. Turtles, butterflies, the lanternfly, you know. I don't know. I'll have to check that. Is it a Megan? Who's MW? I'll need your whole name. Maybe if you can hang around after class, we can take a look and I can find out. I don't know just randomly off the top of my head. And if it was a late submission, I'm not sure that it counts them, how it counts them. So I'll have to check that, because I know some people actually tried to do that quiz after the due date. So it would be helpful if you stayed around and then we could talk. Questions people have? Oh. Thanks. I didn't know that. I set that rule. It's the first time I've set that rule. So that explains it. It shows as your dropped score then if it's lower than the first one. That rule, remember, is only going to apply if you take all of the quizzes. I've already had some people say, I missed the quiz. Can I just count that as my drop quiz? And that's not how it works. So you're required to take all the quizzes. And if you take all of them, then I will drop your lowest one at the end of the semester. So, but I didn't realize it was doing that already. That's what it is. Yep. All right, questions about the content specifically for quiz 3, which the pending doom for tomorrow. So 8, 9, and 10, introduction to metabolism, cellular respiration, and photosynthesis. I do have a guy that you should watch if you want to watch some short YouTube videos that are super content based and are at the level that we need for this class. I always recommend to students to go to Bozeman Science on YouTube. It's BOZEMAN SCIENCE.com. He is an advanced placement high school biology teacher out in Montana, I think, or Wyoming. Bozeman, Montana, right? Yeah, Bozeman, Montana, out in Montana. And he uses Campbell and he's with his students and he's great. He talks super fast because he tries to get them into these short 12 to 15 minute snippets. So you'll have to pause it a lot if it's something that you're really trying to learn from him. But I recommend him all the time because he uses all the language that will show up on our tests and it's the same language that's in our book. And the level of content is what you need for this class, right? So some of the other things that I know you watch as supplements, not to pick on the Amoeba Sisters, but some of that is not detailed enough. It's a great place to start, but it's not necessarily enough detail for you to do well on our exams. So that's just a little tip if you want to watch somebody else. You're tired of listening to me. Yes, there'll be multiple choice. There might be some matching on this one. I'm not sure. I think so.
Are there any topics that we should really focus on? Because on the last exam, I focused on like some certain topics and there weren't many questions on that and it like messed me up.
Yeah, that's gonna happen because you can't like, know what's going to exactly be on the test. And the first, some of everything that we talked about, I mean, this is super detailed and you need to know all of those details. And I think there's about the same number of questions, if not exactly the same, maybe like 2 fewer from chapter 8 than 9 and 10. I feel like 9 and 10 have more meat in them than chapter 8. So you might feel like you studied all this stuff. Usually when that happens, it's because a person focuses on the things that they don't understand as well, and those may or may not be, for that particular exam, what we think is the hardest or most detailed part. I think that's human nature. We focus on what we think we don't know and review what we think we do know less. And I think I would use those dynamic study modules in mastering, because those dive pretty deep and help you kind of sort out what it is that you think you know and you actually know, and what it is you think you know that you don't know. So I would try to use some of those. I've heard from students, like, I don't use them to study because obviously, like, I don't have to study this particular stuff, but I study other stuff. But I would use those, and you can find them. I know that some people, after I say this, they say, I don't know where those are. I didn't see those on Canvas. So I'm gonna go into Canvas right now and go into mastering and show you guys where those are. I remember my password. Okay, so if we take a look in here and go to our course and go to My Lab and Mastering. I don't know if my computer's gonna be able to do all this at once. It's already starting to get a little hot. Over here where it says study area, that's where you'll find those. So if you launch the study area, practice tests are in there, figure walkthroughs, they're the same as the ones that, and the BioFlix 3D animations are the same ones that I've been assigning with questions, but if you wanna just watch the video and not go through the assignment, you can do that. And where are they? Maybe it's study by chapter. Now I'm going to be called out on this, right?
I think the dynamic study modules are when you go back to course home on My Lab Mastering.
Course home. Oh, they pulled them out of the study area. And where are they? Oh, here they are. Yeah. Is that where you guys see them as well? That's reporting. And oh, there they are. I see them.
For us, it's in the top left corner.
Okay. I don't know if they give me a student view in here. But let's take a look at one like for chapter 10. Okay. So they ask you questions, and I'm going to answer this one incorrectly on purpose. Plants use carbon from, I'll just say from the soil, right? Once if you're unsure, click. Twice if you're sure, continue. All right, so I'm just gonna, I'm unsure. Okay, you're unsure and incorrect. And so I thought it sends you, now it's gonna give me another question about the basics of photosynthesis. Does it not send you, I thought it sent me to where to read about it. On chloroplasts, pigments absorb light. Their electrons become excited. Control systems, the source of structures. I think I can't help it. It's hard to not. The most important role of those. Let's do that. So eventually, has anyone gone through these all the way to the end? Eventually, there we go. Eventually, it will give you more data as you make more incorrect choices, and it will help you figure out, like I said, what exactly you know and what you don't know. I like it because these are, they're super, I find them to be the depth and detail that you will need to know for the exam. Right, the light reactions of photosynthesis by the Calvin cycle with, let's pretend we don't know. Let's say this, I'm sure. It says incorrect. I thought that so this doesn't send you back. I thought it sent you back to places to read. Has anybody done these routinely?
I've never seen it show you where it is in the book. When I get it wrong, I usually have to like look through the chapter.
Okay. I guess the important information is all of this, right? How many of them you did know for sure, how many of them you didn't know. And then, like I said, the main advantage is you can really figure out what you know and what you don't know. I would spend some time with those because I think they're good, especially after you've studied. I think they're good to help you. So you didn't see you guys, the rest of you can see, you saw my screen, right? It might be your device if you can't see the screen because it says I'm screen sharing. So you should be able to see this screen right here. All right, so other questions that you have? So if that happened for you guys, it's over here. So when you actually look at the home page for mastering and you click on dynamic study modules, then it will take you to those, right? And I'm using them as independent study. I'm not assigning them. Some people actually use those as assignments. All right, I'm gonna back out of there. Other questions, worries, comments, concerns for the pending exam?
For chapter 8, the notes towards the end under inhibitors, it says reversible or irreversible.
Irreversible,
Irreversible,
IRR.
yeah. Could we go over that?
Sure, so it's not super complicated. It's exactly what it sounds like, right? So an inhibitor is something that's going to prevent the enzyme from working, and it can either be a temporary stop or it can be a permanent stop. Temporary is going to be reversible, right? Permanent will be irreversible. And then those inhibitors, there are two different categories of how they work, right? So all the inhibitors inhibit or prevent the enzyme activity, right? Now, there's two different types how the inhibitor inhibits or prevents the activity, let's say, right? How it does that, there's two categories for that. The first category is over here. Let's say that this is for the irreversible, or let's say reversible. Sorry. Competitive. Competitive or non-competitive. Right? Competitive inhibitors, whoops, function at the active site. So a competitive inhibitor, if I had drawn my enzyme here, right, and that's my enzyme, the protein portion. Oh, and let's put a little zinc back here and as a little bit of a cofactor. And then let's say my substrate needs to fit in this spot, right? So this is the active site where my substrate fits. So if this green blob here that I've drawn is the substrate, then a competitive inhibitor has to be, and I'll make it blue, it has to be almost exactly the same shape, size, and charge, but it's not the same molecule. So it's going to have something else attached to it, or the first portion of it will fit into the active site, but then maybe it has like a triangle piece that sticks out like this, right? So this would fit in there, but it's not going to fit perfectly. That's a competitive inhibitor. It's competing with the substrate for the active site. Now, those can be permanent or temporary. So a permanent competitive inhibitor would be an irreversible competitive inhibitor. Can't take it out, enzyme no longer works, cell will die, or at least this metabolic process will not continue. I'm going to close this, all the mastering stuff, because my computer's not liking that very much. All right, let's see if I can get back to my sharing. Sorry. I lost my whiteboard there. Here it is. So competitive reversible would be, an example of that would be usually like a medication or a pharmaceutical that inhibits a particular enzyme, but it only works as long as you're taking the drug. When you're not taking the drug anymore, then that enzyme is going to go back to its normal activity. An irreversible competitive inhibitor would be, there's a particular antibiotic that's a competitive irreversible, and once it binds, it kills the bacteria. Those apply for non-competitive inhibitors as well. Non-competitive inhibitors just work a little bit differently. So I'm going to draw my enzyme again over here. A non-competitive inhibitor, so if this is my protein portion of my enzyme, I'm not doing such a great job of that. Let's do it like that. This. And my substrate fits here. This is a little slightly different substrate, right? Because it has a little bit different shape. Get around on the outside like that. For a non-competitive inhibitor, that happens when we call those allosteric inhibitors. They bind someplace else. So I'm going to redraw my enzyme here and try to leave the same shape of a hole. A non-competitive inhibitor binds back here and actually changes the shape of the active site. So this red thing that I'm drawing, that's the inhibitor, and it's going to change the shape of the active site. Let's say it makes it look like this. So now my substrate that was this blob here, right, with its very careful little indentations of positive charge and negative charge that's supposed to fit in there, clearly it can no longer fit in there, right? Can't fit, it's too big now because the active site shape has been changed. So this is a non-competitive inhibitor, an allosteric inhibitor, they're called actually, because they change the stoichiometry, not stoichiometry, they change the shape of the active site. Excuse me. This, just like competitive inhibitors, this can be reversible, meaning it can come off. And the example we talked about extensively is a feedback loop or an end product inhibition. And you can look at those, excuse me, in your book or watch that part of the video again. Or they can be permanent. And then if they're permanent, this enzyme will no longer function. And again, if this happens to lots of enzymes in the same cell, then that cell's going to die. Is that good, Leah? Do you need more than that?
No, that was perfect. And for a substrate to fit into an active site, the shape, size, and charge must be correct?
Yes.
Okay.
Correct. And I didn't do a bang up job of that with my mouse here. But yes, the substrate should be the same shape, the same size, and the charge, by the charges, we mean here, let me get yellow, I know it won't show very well, but right here along the interface between the substrate and the enzyme, that's where the charges have to match particularly. So if, for example, there's lots of amino acids here that all have positive charges, then that portion of the substrate has to have negative charges there so that it'll complement and bind. Does that make sense?
Yes, thank you.
Yep, you're welcome. Oh, excuse me, guys, I've blown my nose. All right, other questions for the pending doom? No? Okay, so this one's 8, 9, and 10. I'm pretty sure it is all multiple choice. If it's not, there might be matching or true false. There's not fill in the blanks because I don't know what your experience has been with Canvas, but it's really difficult with science classes because Canvas, if you put an S on a word or if you make the first letter capital and I type it in as lowercase, I can type in all the permutations, but if I don't think of something that you're going to actually do, then it marks it wrong and I have to go through and grade them all by hand. And in a science class, because there's a lot of spelling and it has to be correct, that poses, a lot of problems. So I don't use fill in the blanks in Bio 1. In micro and genetics, in more advanced courses, I feel more comfortable using that. But for our purposes, sadly, your spelling will just have to, of science words, will just have to stay in whatever state that it's in until you get to anatomy and physiology, and then you can worry about spelling all your words correctly. All right. Okay, if anyone needs to hang around after class for a particular issue that I didn't cover today, please do. Everyone else, good luck tomorrow. Just focus, take your time, study as much as you can beforehand. Don't panic. I know exams are stressful. They're stressful for everyone. Overall, as a class, you guys are doing great. There's lots of people that are getting A's and B's and lots of people that are in the C range that, might move one way or the other based on the next few exams. So don't give up. This is probably the most difficult test, I think, in the whole class, maybe. I think I'm scared too. Try not to be a little bit scared is good because, right, it gives us our flight or fight response, gives us a little bit of focus and nervous energy. Too scared is bad, right? Hang around and ask more questions if you need to, right? Because I have office hours for the next 2 hours. So just if there's something else that you want to ask me specifically, just hang around. It's no problem. All right.